Alright, good afternoon everyone. Over the next 10 minutes we'll be talking to you about CRISPR-Cas9 and its applications to viral resistance in plants. Before we get to it, let's look at why we need CRISPR-Cas9 in the first place. The current world population stands at above 7.5 billion people. This is the result of a 60% increase in just the last 20 years alone. If current estimates are correct, the global population will grow at around 1.13% per annum.
These growth rates mean that our population will rise to above 9.3 billion individuals by the year 2050. It's estimated that global food usage will need to double in order to support this population. Even now, our food production rates are failing to rise with the population. For example, available cropland per capita has already decreased by 50% in the last 65 years. By 2100, it's anticipated that our numbers will rise to well over 10.1 billion individuals. As it can be seen, food security is a massive issue for our society.
Given the vital role plants play in providing for our world food supply, it seems necessary that we should examine the threats to agricultural output per year. Plants can experience stress in two main ways: abiotic and biotic. Abiotic stresses involve things like heat, drought, and natural disasters, whereas biotic stresses tend to involve insect pests, bacterial pathogens, and phytopathogenic viruses, or plant viruses, which we'll focus on now. Phytopathogenic viruses alone are known to account for a 10 to 15% loss in agricultural output per year. Annually, this viral destruction is estimated to cost the global economy about $30 billion. What's more is that viral epidemics are currently very difficult to manage.
So if we're considering that things are very difficult to manage, we need to consider what has been done about it so far. So current approaches towards viral management in plants have only proven somewhat successful. Historically, management practices have involved vector-based approaches, which include the removal of insect pathogens and preventing transmission events as a result, and culture-based approaches, which include crop rotation and early sowing.
There are also molecular-based approaches that have been adopted, such as RNA interference, as well as breeding-based approaches, such as
multigenic traits are generally connected together, so you can't prescribe them at the same time selectively. So we need to consider problems with these methods and why we can't use them to effectively always manage plant viruses.
First of all, viral infection of plants is often the result of multiple different viral infections. So if you have one plant resistant to one certain type of virus, this may not confer enough resistance to ward off infection.
Some methods of management are time, labor, and cost intensive. This is particularly relevant when we consider that many of our growing regions are in developing nations. These treatment protocols might simply not be feasible.
Another thing to consider is that traits of resistance in plants are often multigenic, making breeding-based approaches near impossible.
There are a number of confounding epidemiological factors associated with viral outbreaks. For one, we can't necessarily predict vector transmission, and therefore the location of outbreaks that might occur. Additionally, viruses evolve quickly in an evolutionary timescale. Therefore, a technique that we may develop today might not work in a certain period of time. Together, these factors make it very difficult to develop effective long-term and economic solutions. New developments in CRISPR-Cas9 technology are proving promising in this area.
Before we look at any examples where CRISPR has been used, let's have a look at what it really is. CRISPR-Cas9 is a tool used in editing genomes, which allows for specific removal, addition, and editing of targeted sequences. It is comprised of two main components, Cas9 and guide RNA. Cas9 is an endonuclease derived from Streptococcus pyogenes, which cuts specific targeted sequences within the genome of interest. Guide RNA, on the other hand, is a scaffold sequence of RNA, which also contains target sequences of about 20 bases. This binds to a specific target DNA sequence and guides Cas9 protein to cut at the correct location, which induces double-strand breaks and mutation upon repair.
So how is it used in plants?
Firstly, we're going to have a look at an example of Arabidopsis thaliana and its resistance to turnip mosaic virus. CRISPR-Cas9 was used in this study to target the EIF-ISO4E coding sequence in Arabidopsis. There are a couple of things we need to note here about this study. Firstly, Arabidopsis contains both paralogs of EIF-ISO4E. The other paralog is EIF4E. There is a genetic redundancy here, as Arabidopsis can grow and exist naturally with only one paralog functioning. The second thing we need to note here is that the proteviral VPG, or viral protein genome-linked protein, of turnip mosaic virus binds only to EIF-ISO4E, which is a step necessary in the exploitation of host translational machinery. This study targeted the 5' region of the open reading frame of EIF ISO 4E, as it was believed that would be greater chances of frame shift and early stop codon mutations, resulting in non-functional protein being translated. This is just an overview of the method. So first of all, Arabidopsis plants were transformed by floral dipping containing Cas9 and the guide RNA, as well as BASTA resistance gene. BASTA is a herbicide. Next, transformed plants were selected for by spraying with BASTA. This killed off any non-transformed plants. Following this, a T7 assay was used to identify plants with a mutated EIF ISO 4E mutation, indicating active CRISPR-Cas9 activity. Following this, a transgenic plant displaying clear signs of EIF ISO 4E mutation was used to produce subsequent populations via cell fertilization. Finally, inoculation of fluorescently tagged TUMB was carried out upon EIF ISO 4E mutants, the wild-type Arabidopsis plants, and a previously published mutant of EIF ISO 4E. The results of this study found that in the systemic leaf samples of EIF ISO 4E, no traces of TUMB was detected, versus TUMB being detected in all wild-type samples. This is illustrated in the picture in the top right-hand corner. The green segments of leaf are indicative of viral infection, as viruses were tagged with a green fluorescent protein. As you can see, the wild-type sample contained viral infection, whereas all the mutant plants were free of it. Another thing to note here is that growth of EIF ISO 4E mutants was found to be insignificantly different to the wild-type. So not only can you confer viral resistance in Arabidopsis, but you won't inhibit its ability to grow.
So the next thing we could talk about was CRISPR-Cas9 mediated resistance to tomato yellow leaf curl virus. So in this case, both the open reading frames and highly conserved intergenic regions of this virus can be targeted using CRISPR-Cas9 technology. So the scientists involved in this investigation developed single guide RNA molecules for the virus, which were then integrated into a tobacco rattle leaf virus host. The single guide RNAs were then introduced to their host plant, expressing high levels of Cas9 endonuclease by agroinfection. 1 week after these initial steps, the plants were exposed to a clonal strain of their pathogen. 10 days after this infection, the genetic material was extracted from their host and examined to determine the effectiveness of the treatment. So this is a graphic explaining the method itself. As you can see on the left, the guide RNA is implanted into its host vector. The vector is moved into its host plant, which is then infected by the virus itself. The tissue is extracted from systemic leaves, and the DNA is examined by techniques such as PCR and the like. So when we look at the results of this study, semi-quantitative PCR showed that viral DNA was less abundant in host plants than that harbored the virus than those that didn't. This can be seen in the graphic to our left here, where while 7 and 8 have highly abundant levels of viral proteins, and our mutants on the left do not. So this is because the intergenic sequence was altered in the viral DNA. This region conserves the highly conserved sequence involved in DNA replication. By mutating this site, the Cas9 makes it inaccessible to the replication proteins. As such, the virus cannot replicate when within its plant host, and any infection is asymptomatic.
So you're probably wondering, why should we care about these results, and what do they mean?
So CRISPR-Cas9 technology may be used in the future to confer viral resistance traits to agriculturally significant plants.
It has also been demonstrated to be broadly applicable and can be utilized to manage many problematic viruses such as turnip mosaic virus and tomato yellow leaf curl virus.
Preliminary investigations that have utilized CRISPR-Cas9 technology to target both transcription machinery and intergenic regions of sequence have shown very promising results.
This technique has also demonstrated potential to help recover crop losses due to pathogenic viruses.
This would prevent up to 10 to 15% of crop losses and save us about $30 billion.
Altogether, this will contribute to an increasing agricultural efficiency and a better food security for our future.